My name is Melody Roelke Parker, and I am a veterinarian and a scientist, presently working at the National Cancer Institute based in Frederick, Maryland. I'm here today to share with you a story of adventure and discovery that I was involved in a number of years ago in the eastern plains of the Serengeti Plains in Africa. And what I'm going to share with you is the journey that I went on to discover a brand new disease, emerging disease in the African lion, canine distemper virus. And before we get started, I just want to thank a few people that contributed a lot to the video presentation and the slides you're going to see today. Specifically, Annie Hilborn helped with the some slide presentation, Ray Hilborn with the video, Craig Packer, Christophe Reitier with a lot of the slides you're going to see, and especially I want to mention the Messerli Foundation in Switzerland, Zurich, Switzerland, who was responsible for the major funding for the field part of my project. So with that, let's go ahead and start with the slides. In the fall of 1992, I had the opportunity to realize a dream come true in that myself and my family went to East Africa to start a field investigation working for the Tanzania National Parks. I was seconded to national parks as a wildlife veterinarian, as their chief veterinary officer, where my husband and I started on the refurbishment of an old veterinary facility that had been used in the late 60s as a world-class facility. And our idea was to set this up as a first-rate field setting, complete with photovoltaic solar panels, like you see here, 80,000 liter water tanks. We gutted and refurbished the entire inside of this building so that researchers from anywhere in the world wanting to come to the Serengeti could come and work in a good environment with water and appropriate electricity. All the materials were all brought in 300 kilometers away, all had to be hand-hewn and worked up into a final setting as you see here. And this was the final lab that my husband was able to develop. The main purpose of my going to Tanzania was to develop a veterinary program for Tanzania National Parks. It had been 20 years since there was any veterinary involvement and any disease investigation in the wildlife of that country. And one of the major questions was trying to document what sort of diseases were considered to be natural, such as this situation here with a buffalo dying of drought, consequences of drought. 17,000 buffalo died in the fall of 1993-94, and as you can see, this animal horribly emaciated. Histopathology and other necropsy findings did not reveal any disease problems, just simple starvation. Often it's very difficult dealing with free-ranging animals to be lucky enough to actually find the carcasses to be able to determine whether you have a natural problem or some disease state. Because of the network of carnivores, scavengers, such as the vultures, the hyenas, often the evidence that we might want to look at is completely removed quickly from that environment. But this is one of the tasks, to set up a sentinel monitoring situation to just keep track of what kind of problems all the animals are having. Another major part of my work was to involve local Tanzanians and people from Morogoro University here in an outreach program of training, and they actually came up and participated in the disease investigation of the African lion. Also importantly, National Parks was very concerned about the health and well-being of animals that various researchers were handling. And so any researcher that wanted to handle their respective study animal for bloodletting or for radiocollaring, I became involved in the medical management, anesthesia, and handling of that respective animal. And these are the lion team, the researchers that I worked with, and became quite involved in their project as we undertook the disease investigation. Now, what actually happened was, for a year and a half, I had been living in the Serengeti dealing with the occasional sick lion, but I had only actually handled a single lion in a year and a half that I was there, prior to January of 1994. And then in January, suddenly it was as though a war broke out among the lions. And all of you who are familiar with the natural history of the lion will appreciate that lions are aggressive. Yes, they fight. Yes, they defend territories. So fight wounds and battle scars are not an abnormal finding whatsoever. However, what was happening in a very short period of time is I was being notified by local village people who found carcasses like this young lioness here, a subadult, who had been brutally battled and killed by what looked to be a major carnivore, most probably a lion. And you can see here that there's no question that this particular lion had its skull crushed You can see the blood here. You can see the distorted skull cap. This animal, on face value, looks like, there's no question, it was killed by aggression. As the story went on, over the next couple weeks, I was dealing with many lions. And here these are canine puncture holes in the skin, penetrating through the abdominal wall into the liver, into the internal organs. Often there would be no sign externally of wounds, but during the necropsy procedure, I could find evidence of severe internal damage. And what became obvious to me in a very short period of time, over about a month, is that I was presented with many, many carcasses of lions that I had not seen before. And I had a hard time believing that this was a natural, normal, behavioral problem in these lions, that they were just fighting with each other. But perhaps something else was going on, stimulating all this aggression. Along about the end of January, I then started seeing animals that appeared to have overt signs of what looked at the time to be immunosuppression. They had gross infections where otherwise you'd have a bite wound and there would be abscess and maybe the animal would get over it. You see here that we have gross necrosis of the tissue, ablation of the scrotum. This was a male. Hideous wounds that were not healing properly. And this animal was euthanized. Then we entered a stage of starvation and cachexia, and in the face of plenty, in the Serengeti plains, 400,000 zebra migrate down into the plains, close to 1 million wildebeest, and that's when this problem was presenting itself. And yet, you can appreciate how starved and emaciated this lioness looks with her ribs showing, and yet she's sitting on a zebra carcass. So something very strange was going on in that lion population. Additionally, we started seeing animals that were taking inappropriate prey. Porcupines are fun, and these are perfectly healthy lionesses here playing with this porcupine. They occasionally take it. It's not a preferred prey item, but what we started seeing was many, many lions were actually taking and killing porcupines, complete with porcupine quills down their throat, passing solid porcupine quills in their feces. I don't know how they did that, but it was a behavioral change. So either they weren't capable of taking the larger prey or they weren't appropriately thinking about it. Behavioral changes. Lions started showing up on doorsteps of villages, lingering outside of cars. Yes, they're tame in the Serengeti, but not this tame. This was not appropriate behavior. And you can see this lion here right up against a building in the tourist center. Not appropriate behavior. Then one day I got a break, and you have to appreciate that when you're in the midst of a disease epidemic that I didn't even know I was in at the time, I was just seeing lions killing each other, and we had all this aggression going on. All right, then, at about a month later, now we're early February, some tourists on a balloon ride were watching this magnificent lion. They've come to Africa. They have a chance to see their first male lion in the flesh, and there he was sitting there so regally, arching his head, and all of a sudden, on camera, in front of them, this animal leaped up, flipped over, and had a grand mal seizure right in front of them, and quite to their horror. And I was able to contact these people and find out where this lion was and actually go out and view the animal for myself. And now I want to show you what this actually looks like, what this animal was going through. So we'll look at a videotape. And this is this particular lion, and he's in the beginning stages of a seizure. And what you can see is he's starting to raise his head slowly, slowly. All right, and now you're going to see his jaw start to move. And this is a very typical Jacksonian march presentation of a grand mal seizure. And you'll see that this will just spread throughout his facial muscles and end up going out through the whole body. I watched this animal for several hours, and he was having one of these seizures about every 15 minutes. I should note that the odd little sound you hear is my 6-month-old baby daughter in the car with me, so it's not the lion. I learned later that this lion was actually, some tourists had seen him 36 hours before, so he was going through this for many, many, many hours. And by this point, he's just so exhausted, he can't even put full-blown effort into it. Each seizure lasted about 45 seconds. in the actual grand mal part of it. If you watch his pupils, they're maximally dilated. He was cyanotic, pretty bad shape. So I hope you can appreciate what this animal went through if having these seizures every 15 minutes. So given his condition, it was not surprising at all that this animal died. Now, whether or not he actually died spontaneously or was killed and eaten by hyenas, I'll never know. But by morning, this is what I was left with. And at least I have to thank the hyenas for not running off with the skull, because you can appreciate that an animal having grand mal seizures, the brain would be a very important structure to take a look at. And again, because of the carnivore scavenger system we have there, I was very lucky to get anything. And unfortunately, this particular animal did not reveal the true cause of the problem, at least not initially. And that was because he'd been having seizures for so many hours that the pathologists, when they initially looked at his brain tissue, all they found was polioencephalomalacia because of the anoxia that had been going on for all that period of time. And the true nature of the disease process was not recognized. So we continued. And about 3 weeks later, after this particular first episode, so now put in perspective, we have a period of time where animals there's a lot of aggression and a lot of fighting going on. And then that seemed to die down, and we started having neurologic presentation, at least the one case. At that point, I did not relate them at all to each other. They were just spurious events. This, however, one day I was viewing lions, and I saw, if you can watch this, what I saw watching this lioness, watch her face. And this is not motion picture, but you can see a very pronounced myoclonus of the facial muscles. And the moment I saw this, I knew that we were dealing with a neurologic disease process. We had the grand mal seizure, we have myoclonus, and then we had some very, very interesting eyewitness accounts from tourists who had actually seen a perfectly normal looking lion walking along with his male partner, go into a grand mal seizure, and his partner attacked him and brutally wounded him. So now I have linkage of the neurologic signs with the previous aggressive aggression mortality. And it looks like we're dealing with one large problem. About that time, also, radiotelemetry cats started dying. And this became very problematic for working on this disease problem, because this is what I normally was able to collect from animals that died, is nothing but an empty radio collar. And it's very difficult to do a necropsy on that. You might get a few hairs or a spot of blood, but there's not a lot of information. So in the first few months of the year, this was the presentation. We had observations of a number of animals having grand mal seizures, also based on tourist observations. We had several animals with myoclonus, and I had the carcasses primarily from the aggressive deaths. And as a good budding pathologist, tissues were taken and stuffed in the formalin jar, even though at the time it was perfectly obvious that the reason they died was because of aggression. But tissues, nevertheless, were saved and preserved so that, luckily in this case, we could go back and take a look at those tissues as a potential source of disease problems. Also at that time, we started working with national parks and got permission to handle perfectly healthy-looking lions. And this was not an easy task, given the political climate in a number of national parks where researchers are sometimes viewed as problematic in terms of animal survival. And so we had to work very closely with national parks to allow us to actually go out and handle perfectly normal animals. And in the Serengeti, we would dart these animals using a telangiect dart system, a very lightweight plastic dart, using tilazol or ketamine Valium combinations, and proceed to get blood samples from both animals that were clinically affected as well as those that seemed to be healthy, because I wanted to develop a profile of the clinical picture of this disease. And you can appreciate here, this lion is in very poor shape, extremely emaciated, bony hips, ribs, and you might be able to tell here I have an IV fluid bag hanging. Our aim was not in any way to treat individual lions in the Serengeti. There are 3,000 lions in that ecosystem, but I did not want to be responsible for the death of a single animal during an anesthetic event. And so if an animal was in bad shape, like this animal, they did receive extra special care to get them through that anesthetic event so that we could get our samples. Some of the clinical signs that I noted is many of the animals had lymphadenopathy, particularly noted in the axillary lymph nodes, 5 centimeter long, 3 centimeters high, a couple centimeters wide, very pronounced lymph nodes. Also, the popliteal nodes were markedly enlarged. Another very interesting clinical finding was that many of the animals that didn't demonstrate myoclonus in the awake state, I could elicit evidence of neurologic damage when they were asleep. And granted, this animal is under anesthesia, which is not the most appropriate way to do a neurologic exam, I'll grant you, but given the situation of that lion with those monster teeth, this is what I had to deal with. Anyway, what I saw in these lions was that if I was to touch the vibrissae of this particular lioness, I could elicit a palpebral response, which is not typical of how those facial nerves normally behave. And in fact, you can see here where I've drawn out the pattern where if I touched anywhere within above that black line, I could elicit a palpebral response. So here, for instance, if I touch way down here, the eye would still blink. So even though this animal looked perfectly clinically normal, there was evidence of neurologic disease. And additionally, every single animal that was handled, we took a great number of biological samples. And I think this is very critical. Anytime you're handling a free-ranging animal and you take the risk of potentially damaging that animal or worse, killing it, I feel it's our professional responsibility to learn as much as we can from that animal in terms of taking lots of different samples, maybe far more than what you personally need for your own specific study. And so in this case, we took everything from serum to heparinized whole blood to fecal samples to ectoparasites and the gamut. Many of the samples we banked down for future work, maybe ideas and questions that haven't even occurred to us yet. And here you see we start processing these samples. At this point, this was in my house before our lab was completed. Now, where were we? I've mentioned to you that we had these spurious deaths going on. We've got myoclonus. It certainly looks like a disease such as canine distemper could be at play here, but I had no evidence of that. In terms of what we had going in the whole population, what you can see here is the demographic pattern for the last 20-some years of the lion population in the Serengeti. And this is work that has been carried on by Dr. Craig Packer, starting originally with back in the 1965 by George Schaller. And see here in the last 20-some years, this population does have a lot of ups and downs, highs and lows, but nowhere has it had this precipitous decline of over a third of the population in a very, very short period of time. So some of these earlier declines have been attributed to starvation or drought, but we've never seen this big hit as happened in the spring of 1994. Now, going back to the necropsy specimens, as I've already said, by taking specimens always routinely, preserving them in formalin, even though if you don't immediately see a need for them, but then they're available for retrospective studies, and that's what happened in our case here when we had all these aggressive mortalities. I had the tissues, we were able to send them off, and to find out, look for clues about the histopathologic clues of what was going on. And lucky for me, I had sent a very few number of samples to Dr. Linda Munson, who was at the University of Tennessee, and she had worked previously on an outbreak of canine distemper in the felids, lions and leopards, in an outbreak of canine distemper in California, zoo cats. And so she recognized this nice syncytia, this nice giant cell here, typical of what she found in lions with canine distemper. As soon as we had this kind of evidence, samples were sent immediately to Dr. Max Appel, there we go, at Cornell for immunohistochemistry staining using monoclonal antibodies against canine distemper. And you can see all this rusty stained material here. That's evidence of viral particles in this tissue. So at this point, we needed to confirm that, okay, yes, it looks like we've got histopathologic evidence of a canine distemper-like, at least presentation. We've got monoclonal evidence saying, yes, we've got canine distemper here. But how about serologic evidence? And what we were able to do is send off serum from all the animals I had been handling that were alive, Okay, that's important. The living animals in 1994 that I was able to sample, as well as retrospective samples from animals that were captured for some other means. And you can see here, starting in December of 93 and pulsing in 94, is when we have this rapid seroconversion of the entire population. So basically, we had a naive group of lions in the early 90s, and then when canine distemper entered that population, we have a rapid seroconversion of, by the time it was over, of over 85% of all the animals that I handled. Now, at that point, we still didn't have the virus. Most of the tissues that I had collected were from spontaneously dead animals that had been dead for a varying number of hours, and unfortunately, despite numerous viral isolation attempts, no virus had been isolated. And then one day I got a break in terms of getting access to a young neonate lion having grand mal seizure. And this animal was euthanized, and we collected cerebrospinal fluid. And from this tissue, we were able to grow virus. And Max Appel got the virus out. And this is the only lion to date that we actually have virus from. In contrast, many of the other species that I'll talk about in a little bit, bat-eared fox, hyena, had abundant virus, and it was very easy to grow that virus in a culture setting. So once we had the virus out, Dr. Margaret Carpenter at our lab at the National Cancer Institute started looking at some of the genetic profiles of a couple genes, the P gene for one, and the H gene, and tried to look at what was this virus. It looked like it was a canine distemper-like organism, but were we potentially dealing with a situation like the phocids or the dolphins, where they had their own unique type of virus, that maybe it was just cross-reacting with the various monoclonal antibodies? And so through Margaret's work, she was able to sequence this gene and discovered that, in fact, 2 isolates of the Serengeti population here were, in fact, very closely aligned to the Davis-Thompson strain of the canine distemper virus. And this is the vaccine strain. And this basically is just a tree. The closer you are together on this tree, the more closely you are in evolutionary history. So you have the phocid virus here, the canine distemper cluster, far away from the dolphins, measles, and rinderpest virus. Okay? So at this point, we pretty much were in conclusion that we were dealing with a canine distemper organism that, for some reason, had suddenly gotten into lions and was causing pathology for those animals. Now, let me digress a little bit. Going back into the 50s at least, it has been known that cats can be infected with canine distemper virus. This is work that Max Appel did a long time ago, and I may have the date wrong, but long ago he infected domestic cats, and in fact they seroconverted to canine distemper virus. However, there was never any pathology seen with domestic cats. And up until the early 80s, it was felt that among the felids, canine distemper was not a problem. And then in a zoo where I was working in Western Oregon, I diagnosed canine distemper in a Bengal tiger. And that animal also presented initially with myoclonus and progressed over a year and a half to the point that we euthanized her, and she had full-blown dog-style canine distemper. And at that time, there were a number of other papers published that demonstrated that tigers were susceptible to canine distemper, but that it looked like they were tigers that were in peculiar situations with dogs. They were circus cats, they were people's pets, and the like. So it seemed not to be a very critically important problem for the felids. And then in 1991, unbeknownst to me, is when this outbreak occurred in the western states in California among leopards and lions in a zoo setting where they died of canine distemper. And I was in Africa by that time, so I was unaware of this particular problem. So I then was having to deal with canine distemper in the lion de novo, unbeknownst to me. So where do we think it came from? Well, a logical choice in that part of the world is the domestic dog, because at least based on the genetics we've done so far, it is a domestic dog virus, and we have ample 30,000 domestic dogs in the surrounding villages in that ecosystem. And it is known that yearly there are epizootics of various diseases that go through these dog communities, knocking them down, so that the mean age of the dogs are 2, 3, 4 years old. They're not an old dog population. So luckily for me, at the time that I was there was another graduate student, Dr. Sarah Cleveland, working on dogs. Her primary question was looking at rabies, but we asked the question about canine distemper. And you can see here, this is the area where the dog density is very high. Every single year, over a 3-year period, pups of the year had a prevalence of around 50% of the animals were seropositive for canine distemper, showing that this virus is ubiquitous in potential potential for jumping out of the domestic dog. Some of the other communities, such in the Maasai community, it was there, but in much lower number. The prevalence was much, much lower. Oh, excuse me. In earlier years was much lower, and then in the epidemic year, it jumped higher. Other species that I encountered in the Serengeti that had canine distemper was the bat-eared fox. And what I found fascinating was that each of these species were, when they caught this virus, they behaved in a completely different fashion. The clinical presentation was very different. The fox appeared to be the most susceptible, and you see here oculonasal discharge pronounced to the point that his eyes were completely glued shut. He had diarrhea, pneumonia, seizures simultaneously, and this virus seemed to sweep very rapidly through the fox dens that were affected. And in fact, I was lucky in that there's a filming company, Hugo van Lauwek's company down on the southern part of the park, and only because they were out there viewing some of these dens were we able to document a fox actually having a seizure, and they notified me of this particular problem. And I went down 3 days later, was able to dig out the den series, the den, and actually find one of the dead foxes that had gone down in this burrow and died. And we were able to get virus out of this animal. And I just want to mention that to you, when you work with free-ranging animals, you take specimens wherever you can. And even though you might think, as a good pathologist, that no, we're not going to touch this animal because it's been dead for 3 days, this may be the only thing you have to work with, and you should take everything you can. Other species, we have a leopard here that was actually killed by a lion. And although this animal was a number of days dead by the time I did the necropsy, there is faint histopathologic evidence that this animal also had canine distemper. A safari lodge, renowned for its large population of civet cats that would come and sit in the rafters in the evenings as you drank your sundowners, all disappeared. 23 animals disappeared in about a 2-week period. No bodies were ever seen or collected. Just simultaneous with the disease epidemic in the lions and the bat-eared fox, these civet cats disappeared. Hyenas, another species thought to be resistant to canine distemper virus. I had a number of animals, mostly juveniles, that presented with signs of canine distemper that looked much more like a dog. And you see this animal is alive, extremely dehydrated. He had a fever of 107, 10% plus dehydration, oculonasal discharge, pneumonia. No signs of neurologic disease were documented in the hyena, at least by me. So again, different presentations in the different species. Interestingly, the lions never showed any of the early signs that are typical of distemper, such as oculonasal discharge, pneumonia, or diarrhea. It's as though they skipped that and went straight to the neurologic form and whatever the starvation cachexia problem was. Again, just another example of tissues where you can get them. This animal had been obviously highly scavenged, and yet right down there was a nice mandibular lymph node that the vultures had neglected, and we were able to document that this animal also died of canine distemper based on histology of that lymph node. Now, looking at genetic profiles of all the different isolates that we were able to obtain from the Serengeti system, what we see, again, this is based on the H gene sequence, the fox, the hyena, the domestic dog, and the lion, basically, with one minor change, are virtually identical on this tree, indicating to us that this virus is freely jumping species to species to species in that system. And we think that the hyena is probably a very, very important animal in that ecosystem for the spread of this virus, because Although I already stated we think this virus probably somehow got from a domestic dog, into the lion population, it didn't get there directly. And the most probable route that it came is through other species, such as the hyena, which have tremendous commuting routes that they take, 100 kilometers out in each direction, following migratory prey, going out into local villages where they could easily encounter domestic dogs and bring that virus back into the park. That is yet to be proven, but it's certainly possible given the natural history of that species. Now, where are we with the lion today? This virus, canine distemper, came through that population in the spring of 1994. Basically, a third of the population died in a 4-month period. However, every animal that survived that epidemic and has seroconverted, we believe, is basically protected for life. This virus does not appear to have any long-term reproductive impact, so that the lions are in fact reproducing and doing well. So in the Serengeti system, even though, yes, 1,000 animals probably died, we don't know for sure, and yes, that sounds like a pretty horrific number, in terms of long-term impact on this population, it's probably fairly minimal. What is very important to us in terms of documenting this is the apparent shift in the pathogenicity of this virus. And we say that because going back pre-1990, going back to bank samples back to 1985, we have zero evidence that canine distemper was in lions earlier. And if you time point from the age structure of the animals that were seropositive in 1985-87, it looks like canine distemper was last in the lion population in the early 80s. Then you've got a 14-year hiatus before it hits again. And what's interesting is if we go back and look at that demographic profile of the lion population in the early 80s, there's no diminution of population numbers. And the other intriguing evidence that suggests this virus has been in that lion population for a number of years goes back to 1965 with George Schaller's comment in his book, The Serengeti Lion, of 2 lions with neurologic twitches to the face, which, given what I see with the myoclonus of the facial muscles, I think is almost pathognomonic to suggest that canine distemper has been in these lions for at least 30 years. And yet no one has ever seen the kind of mortality that we saw in 1994. So whether there's been a shift in the pathogenesis of this virus or whether other ecological factors were involved are yet to be discovered. Some interesting questions that we still have is, given these seroprevalence, these are prides, each circle is a pride. Only one pride right here in the middle was the only pride that had no evidence of seroconversion. And all these others there's about 5 or 10 animals in each pride that I sampled. So despite that all these animals appeared to have exposure to the virus, only certain prides had very high mortality with animals looking like this. Let me just back up a moment. And those prides were up here in the northern part of the range. This is where we had mortality of 50, 60, 70% in some cases. And this is where the animals exhibited this profound cachexia, like you see here. Whereas prides to the south, and that's where the red stars are, this is where we had disease. Down here, I know the virus was there and active while I was doing the study because I had documented seroconversion in 2 lions in this pride right here in the face of cats that looked like this. Babies were on the ground, no mortality to juveniles whatsoever, and yet that virus was circulating at the time this photograph was taken. So we have some big questions about why. Why didn't it hurt and kill certain animals and others? One very important question we need to still undertake is it possible that the virus was differentially pathogenic just in the virus itself between what was circulating in one pride versus the other? So that's an open question. Another aspect we're currently investigating is such things as the MHC of different prides of lions. Now, lion prides are maternally linked, so that every animal within that pride has a historic genetic background, going back to the grandmother or the great-grandmother. So it's possible that you've got host factors that are influencing disease resistance among the different prides and the sister prides. And so we're investigating that aspect. Another thing that I personally am involved in is another co-infection with the virus, feline immunodeficiency virus. And in the Serengeti system, a number of years ago, we documented the fact that virtually 100% of all lions greater than 2 to 3 years of age are infected with feline immunodeficiency virus. And we have been following individuals to try to discern whether or not there's any impact of that virus on their immune system, like is seen in the domestic cat. It is sort of conventional lore has come to believe that this virus doesn't seem to be doing anything to them because you do not see chronically ill or affected lions. However, in my experience dealing with free-ranging animals, the chance of coming upon and finding a carcass of a chronically ill, debilitated lion is probably nil. So what the tack that I chose to use was to get samples from clinically normal-looking animals and look at some of the immunological profiles that might give us a hint about whether or not this virus is doing anything to these lions. And basically, by collecting whole blood from the animal and then separating it over a gradient, in this case using histopaque, post-centrifugation you get a nice band of your peripheral blood mononuclear cells here, harvesting those, freezing them down, bringing back to a laboratory setting, and then using monoclonal antibodies that can specifically recognize feline T lymphocytes, and then subset that population into CD4s versus CD8s, cells that are integrally involved in different aspects of the immune system, ask the question of whether or not these lions look like normal, have normal lymphocytic profiles, or whether they are distorted, like is seen in humans with HIV, or cats with FIV. And for instance, this is a situation here of a normal captive lion, and you see the CD4s, 46% of the T lymphocytes versus CD8s, 18%, which is a pretty typical 2-to-1 ratio that's seen in normal, healthy humans as well as cats. Now, asking that same question of my free-ranging African lions, what I saw was a very different profile. And looking just at the CD4 cells, what I have demonstrated is that these are my wild lions here, all of which are positive for FIV. This group over here are captive lions, all negative for FIV, and you can see a marked difference in the values here of the CD4, CD8s, highly significant p-values. Now, put this in perspective, humans with HIV that are breaking with AIDS, 200 CD4s is the cutoff for when you have definition of clinical AIDS, and you can see that these lions are all very, very low. Now, one question that is still to be determined here, is this simply a function of them being wild lions, being exposed to various parasites and other pathogens, such as canine distemper, which we know can cause a lymphopenia, or is this in fact due solely to FIV? By looking at the time frame on which I sampled these animals, a number of them, yes, were sampled during the canine distemper outbreak. However, several of these low animals were sampled a year or 2 years afterwards. So it doesn't look to me like canine distemper is the reason for those low number of lymphocytes. So we are still investigating the potential effect of FIV, and if in fact it is responsible for lowering the lymphocyte populations of these animals, that may make them much more susceptible to a virus such as canine distemper that comes in on the back of FIV. Now, we think that the FIV in the lions has been there for many, many hundreds, if not thousands of years. So we have yet to fully investigate this. But in terms of the lions today in the Serengeti, they seem to be doing fine. But I think the power of this investigation and this adventure that I took you on is to show you the breadth of what veterinary medicine and histopathology and field ecology working together can pull together a whole story about a particular disease problem, and how hopefully this information can then be applied to other more endangered species of cat or even of lion, such as down in the Etosha Pan in Namibia, where you only have 350 animals left in the country, where given what we now know about canine distemper and its potential for causing high mortality in lions, you might consider such things as intervention of vaccination programs and the like in small isolated populations. Thank you.